All right, so today we're going to look at our trace evidence, or at least start our trace evidence lecture. And you'll notice here in the kind of opening slide, if you will, there's six different topics, at least, that we're going to discuss as we make our way through this particular section. And I think what I'm going to do is break these up. So, for example, I'm going to lecture first on the hair portion of these notes, and then transition into fiber, pollen, so on and so forth. A couple of them may be paired together because the sections themselves I don't go into tremendous amounts of detail on. The hair stuff I do, probably a little more so than some of the others, just because the information is something that you see pretty frequently on TV or you read about in the newspaper or online or whatever. So again, to help kind of maybe clear up any misunderstandings, or at least give you as much information as possible to help you understand what that topic might involve. So I think that's the way I'll do this particular section, so we can kind of use that as we make our way through. All right. So as you look at the learning objectives, make sure you can identify the three major parts of the hair, especially when you're looking at the medulla, cortex, and cuticle. Those are going to be the three that you really need to be able to identify and know where they're located. You'll see that on a couple different assessments that you'll have as you make your way through the remainder of this of the course, and then obviously the value associated with each of those. You might want to even throw into that the hair bulb, because obviously when we start talking about forensic value or testability, that's going to be something that's going to be very, very important to us. Identifying similarities and differences between human and other animal hair. So again, certain things that are going to be identified. Specifically the medullary index and looking at the medulla itself, you'll notice a couple big differences there as we kind of make our way through that section. Being able to determine approximately how long ago an individual may have been exposed to a chemical based on the rate at which we know hair grows for most individuals. So you can use that to kind of work backwards, if you will, to end up being able to determine when an individual was exposed to a certain chemical. Or maybe if you're looking at a hair color situation, or an individual that has hair coloring, um, and maybe they were found deceased or whatever, you could figure out how long ago that particular event took place. So the last time that we know that they had been observed alive, or again, when we start talking about being able to link individuals to situations or uh, timed events, being able to know when a particular act took place. So again, we could use that. Maybe they were seen with someone after having their hair done, or getting colored, or whatever the case might be. But again, all of that is going to be important. The fibers and textiles, the differences between them. That section is a little bit long as well. There's a lot of pieces and parts to that. But again, I do try to kind of keep that moving a little bit and make sure we don't get too bogged down just looking at one specific part. Plant material. That one is going to be relatively short-ish. Just because we don't get into a lot of detail with that. Soil sample collection, what we can get from those. Glass, there's a couple different activities we do with glass, so it's important to understand the refractive index in particular is going to be one that's going to be useful. Being able to describe how light changes as it moves from one medium to another. That's one of the ways that glass ends up being analyzed as well. So that becomes very important. And then patent plastic and latent impressions. Again, this is one that we'll see when we do our fingerprint stuff as well, because it's a specific type or specific groups of fingerprint classes as well. So we see a lot of overlap, oftentimes in this section, from one particular topic to another. So hopefully we find that is the case with this as well. All right. So as we look at hair, now I'm going to click on this link just temporarily to kind of show you what we see. But the link will be available in our PowerPoint presentation as well. But you'll notice, obviously, I've linked this to a study from West Virginia University. uh, but very useful in terms of some of the information that it goes over and covers. So you might want to take a look at it. It's kind of interesting, if nothing else, in terms of what you can kind of glean from it. Some of these, obviously, we don't have access to. I don't have any deer hair or mink hair. A dog hair, not a problem. Dogs we've got, and we can access hair from those very easily. Surprisingly enough, we have some goats as well, and so we've got some goat hair that we can analyze as well. Um, but again, when you start looking at some of the specific characteristics associated with them, it becomes very important. So, again, I would encourage you to take a look at those kind of at your leisure, but it's kind of interesting stuff nonetheless. All right. So, as we talk about hair, um, when you look at pieces and parts that are found oftentimes from a forensic standpoint, or things that are shown on a TV show or something like that, hair collection tends to be one of those that is pretty prominent. And there's good reason for that. Hair is very easily transferred from one individual to another, or from a surface to an individual. Right? When we talk about those primary and secondary transfers, hair is a very easily secondary transferred material. If you go visit someone's house and they happen to have a pet indoors, or even a pet that just goes indoors occasionally, it's not uncommon to have a transfer of the hair from that animal onto some article of your clothing, and you may be completely unaware of that. So secondary transfers of hair are going to be very, very common, and obviously I'm talking about it in the context of animals, but the same thing would be true of humans as well. Um, Speaking from personal experience, obviously, if taking even a clean shirt out of the laundry. It's been washed and dried and folded and put in my drawer, and I take it out, and there's a long hair on my shirt. Obviously, I don't have long hair. I don't have hardly any hair, so it doesn't belong to me, and yet it's been able to make its way through the entire laundry process. And so secondary transfers are going to be very, very common when you look at hair evidence. Okay, now hair is going to fall under the category of class evidence the greatest majority of the time, because oftentimes what you're going to be able to ascertain from it is maybe color. You might be able to determine shape and length and things like that. Now, there are going to be some specific characteristics that we'll look at with regard to, like, the medulla and the cuticle and even the cortex. Those will provide some clues as well, but by and large, going to be mostly class evidence. However, we will also talk about its value from an individual evidence standpoint as well. But that's going to be the much less common form of evidence as it pertains to hair. All right, so when we look at it, obviously, hair is designed to help regulate temperature on most organisms that have hair. Now, in humans, that doesn't tend to be the case so much. Our hair is much less dense than what you'll find on most animals. That's why on most animals we're going to refer to it as fur. Uh, but on us, the hair follicles aren't nearly as numerous. There aren't as much, or as there isn't as much hair on humans as what there are on animals. Um, and so we don't have that same density. So temperature regulation on us isn't the same as what it is on most other organisms. The way that's designed to work, we have a muscle that's connected to the hair shaft, or at least the follicle that it's going to grow in, and it will allow that hair to stand upright and help trap air next to the skin of the organism. That's going to serve as an insulator. And then in warm weather environments, the hair lays flat against the skin, so you don't have air trapped, you don't have that insulation, and you don't have that raised body temperature. It's going to be something that's going to be found primarily when it's cool or cold outside, and we experience the same phenomenon. If you're maybe all sweaty from working out, and you step outside and there's a cool breeze, it makes the hairs on your neck or the hairs on your arm stand up. You're like, ooh, kind of chilly. Um, and so that's kind of a throwback, if you will, to kind of what those hairs are designed to do. Um, so anyhow, yeah, that's temperature regulation stuff. And when you look at where our hair follicles are found, again, only 2% are found on our head. We think about them, the greatest majority of our hair follicles being found there, and with good reason. Um, but in the grand scheme of things, there aren't a tremendous numbers that are going to be found in that particular location. So we have about 5 million hair follicles that cover our entire body, and of those 5 million, about 2% of them are going to be found on our head. Okay. All right. So as I mentioned, class evidence, we can identify groups of individuals. We could also exclude groups of individuals, at least possibly. So let's say you find a blonde hair at a crime scene. Individuals that have black hair, provided that it's not been dyed or something like that, could be excluded as a potential suspect based on the information that you're going to gain. Right. Uh, for the most part, hair is going to be class evidence. Now, that's not always the case, right? As you can see on here, unless it has the bulb, if it has the bulb attached to it, then it's going to be individual evidence, or at least it could be individual evidence based on the information that can be gained from it. Okay. We talked about the secondary transfer, primary versus secondary. Again, because hair is very light, it is very, 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 very durable and strong. So it withstands lots of environmental conditions. So it sticks around for a while. And so, again, being able to be transferred from one location to another, from one individual to another location, whatever the case might be, is going to be very, very common. Okay. The first use of hair in a forensic application um, was, they say, took place in 1847 in Paris, and that was the first time that they started analyzing hairs that had been located at a crime scene to be able to determine whether or not a particular individual had been at a location or not. So the use of hair from a forensic standpoint is not new, but the beauty of it is, obviously, as these techniques and our science improve, our ability to get more accurate and better information from hair is also going to improve as well. Now, one of the things that we're going to look at only a little bit, and I say that now because if you go back and look at this hair study, they do a really nice job of showing you the differences that exist between Caucasian, Mongoloid, and Negroid hairs, which is really kind of interesting, just in terms of shapes, but also when you look at the medulla as well, that middle portion of the hair. It ends up being really interesting in terms of this stuff that you can kind of glean from that. So, like I said, if you have a few minutes, it might be worth your time just to take a quick peek at it. All right, so as we look at the structure of hairs, these pictures do a real nice job. Again, giving us a little more probably than what we need, but again, still important. So I had mentioned the erector pili muscle that allows our hairs to stand on end, if you will, when we get cold. And so you can see if this muscle were to contract and shortened. Okay, so as it would pull towards the writing here, it would allow then that hair to go from laying down here, and it would become more upright. So again, doing a better job of at least trying to trap some air to serve as an insulator. You can see the sebaceous gland that's going to help secrete oils that help keep the hair conditioned, allow the hair to be able to grow through the skin. Again, provide that slight lubrication to allow that hair to move within the pore of the skin itself as well. You'll notice the papilla down here is where the blood vessels are going to tie in. So again, because hair is really technically a living part of our body and it grows from the skin, so anything that you and I ingest or take in becomes part of our hair. Now that becomes very useful because we'll talk about a test a little bit later that helps us to be able to determine whether or not certain chemicals are present in hairs, or the quantities of chemicals that are present in hairs. One of the things that forensic scientists can do to see where an individual may or may not have originated from. But anyhow, um, the main parts that we're going to focus our attention on, you'll notice the follicle. All right, the follicle is going to be the part down here. You'll see it's also referred to as the hair bulb itself. But this is going to be the part where the hair is going to grow from, right? And allow those blood vessels then to attach, and they're going to feed that hair, providing it with the nutrients that it needs to be able to grow. All right, so the follicle ends up being very important. That's the attachment point. that allows the hair to be stuck, if you will. And it's not really stuck, but it is stuck of sorts to the skin or in the skin so that it can continue to grow. You've got the shaft, which is this portion, the long portion of the hair itself. That's the part that we end up seeing, and again, we're talking. This would be based on the shape of this and its length. Looks like it might be something maybe on the arm or something like that. But if we're talking about hair growing on the head, it's going to be a lot longer than what we see here for most individuals, or absent in the case of some, such as myself. But regardless, the hairs themselves are made up of keratin. Keratin is the same material that our nails are made of. So it's a very, very durable material, exceptionally strong because it's made up of amino acids, right? And so as a result of the interactions of those amino acids and the bonds that they're going to form, makes that hair really, 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 really strong, which is good. Again, a useful mechanism in terms of protection and of our bodies, reduces friction between different parts of the bodies, things like that. But it's also going to be waterproof and then or provide a waterproof layer and then flexibility. So this is when we think about animals. So think about a dog that swims in the water, right? When they get out and shake off, their outer coat is damp, but not saturated. And if you put your hair underneath that outer coat, the underlying layers, that inner coat of fur tends to be relatively dry, even if they've been swimming for a long period of time. Kind of neat to take a look at that. But that's kind of what they're designed to do. Okay. Now, more specifically to us, so we've talked about the follicle, right? Talked about the shaft of the hair. But when we're talking about these major components, the cortex, the medulla, and then the cuticle, those are going to be very, very important. Those parts of the hair shaft itself are going to be the most useful in terms of being able to differentiate between human hair or other animal hair, and then specifically even amongst the same individuals. So the inner medulla, this oftentimes is going to be a colored portion in the hair. When I say colored, it's going to normally be a dark line that's going to show up. Now, this particular one that's shown in this picture, so you'll notice this one has been fragmented. There's a fragmented is one of the terms that you can see that's going to be associated with that. A continuous, this would just be one thick line. Fragmented, this is the fragmented. Doubled, is also one that you can see where it looks to be there are two sets of dark lines. You can have absent. You can have an interrupted. Interrupted is where this ends up being, each of these little pieces end up being relatively consistent. So again, lots of variations that you'll see, and again, these can be used to differentiate between different individuals. So if you have a continuous, let's say you have a continuous medulla, and you've got a potential suspect, but the potential suspect has an interrupted pattern or a fragmented pattern or something like that, they can probably be excluded because their hair doesn't match, at least in terms of the medulla pattern. Now that's not always true. One of the things we'll talk about is in terms of hair collection and why we have to collect so many, but still very important. Okay, so that's the inner medulla. Sorry, you've then got the cortex. The cortex is the part that actually is the part that contains the color, the melanin that produces the color that we then observe. So this, when you shine a light through hair, again, we're talking about it from a microscopic standpoint. That very intense light allows us to be able to discern the colors that are going to be part of this. Now, we all know that you can change that. So individuals that have colored hair, that dye has been put onto that hair and has been absorbed by that hair to some degree, and so that changes the way that this is going to appear. But again, still useful, obviously from a forensic standpoint. And then the outer cuticle, this is the part that's going to be made up of overlapping scales. And so what you'll see with this, different organisms will have different scale or cuticle patterns. Um, and that then can be used to again discern between different animals, maybe even different species of animals or whatever the case might be. It's going to give a lot more specificity when you're trying to identify a particular source of a hair that you may have collected. Okay. So again, if we go back, we got the medulla, we got the cortex. So medulla is the middle part. Cortex is the colored portion that we're going to oftentimes say is brown hair, black hair, blonde hair, so on and so forth. And then you've got the cuticle, the outermost portion, the protective part, if you will, of the remainder of the hair. Okay. When you start comparing different regions of the body and the appearance of the hairs from those regions. So, the hair locations are generally broken into six different spots on the body. And when you look at those different locations, each of the hairs end up being slightly different. So the six different areas. You've got the head. You've got the eyebrows and then the eyelashes. You've got beard and mustache hair. You have underarm hair, you have auxiliary or body hair, which would be like stuff on your arms and legs, things like that. And then you have your pubic hair. So those are the six different regions. And each of those different regions are going to have slightly specific characteristics. So again, if a hair is recovered from a scene, and the hair is relatively intact, that provides a lot of information to investigators. They're going to be able to examine that hair and be able to determine maybe where that hair might have come from. Okay, but again, there's lots of different pieces and parts. Um, for example, head hair is generally circular or elliptical. Eyebrows are circular, but often have tapered ends. Beards are thick and triangular, so on and so forth. You get the idea. Each of these different body locations, the hairs are going to vary in terms of their appearance and characteristics. Okay. All right. So in terms of collecting hairs, being able to have a correct number is going to be very important. And I say correct number, that's not completely accurate. Uh, but let's say a single hair or three or four hairs are collected from a piece of evidence at a crime scene. That's great, but you have to have something to compare them to. So obviously, very similar to exemplars that we talk about, like in handwriting and things like that. So, most of the time, uh, hair collection is going to not be less than at least 100. And they're going to collect those from all parts of the head because, again, different hairs in different locations, especially if you're talking about someone that may have had their hair dyed or something like that, they're going to have natural variation. And so they're going to be examined to see what characteristics and what traits they have. And then obviously you can use that to determine yes or no, they are similar or not similar to those that were maybe recovered from a crime scene or from an individual or something like that. If you have a suspected sexual assault, 25 pubic hairs are going to be collected again to be able to discern whether or not whatever was present in the evidence is going to match those collected from your potential suspect, right? Again, we talked about the variation that hairs have from person to person, but also even from the same person, you're going to have variances that you'll observe as well. So this gives you your comparators to be able to look at um, as you're conducting your analysis. Different ways to collect hairs from evidence, plucking them. So plucking, you could either so that would maybe you have a suspect and you're obtaining hairs that you're going to compare. That would be a plucking type of event. You're going to pull those whether by hand or using forceps, probably, but plucking from evidence as well. So you're going to maybe examine a coat and around the collar, you're going to see some hairs. So you would take forceps and collect those hairs, stick them obviously in a small envelope, label them, so on and so forth, kind of like we've talked previously. Shaking things out. So again, oftentimes in a clean lab type of setting, you would take a piece of white paper. Maybe you've got an article of clothing or something. You're going to shake that article of clothing. And again, you would do one at a time. So let's say I've got a shirt, some socks, a pants, so on and so forth. You're going to do each of those individually to be able to see what you would get. right? And again, before you shake them, you're going to inspect them very closely, maybe use magnifying glass, alternate light sources, whatever, to be able to see whether or not there's anything there that you would want to get. Scraping, again, you can take a hard-sided object and scrape hairs off to be collected. Taping is where you would take tape and literally go through and touch it to a surface, trying to see if there was anything there that would stick to it. Hair evidence, obviously, in particular. And then if you have a really large area, you can also vacuum that area. Obviously, in that type of setting, most of the time these labs are going to have a very specific vacuum that will allow them to change out filters and bags, so that the only thing that would be used in that vacuum, the only thing that would be collected from that bag vacuum would be the bag from that piece of evidence. Maybe it's a blanket or a bed or an area of carpet or whatever the case might be. Would be very, very useful. Okay. So again, once you have them, you could collect those. You're going to label those. Now, obviously, this is not an acceptable piece of labeling. You would want to be able to know who collected it, when it was collected, where it was collected, from what location of the body was it collected. You get the idea. All of those are going to be very, very important, so that you have a good set of information. So you've got your chain of custody and everything else that obviously needs to follow, so that it would be admissible in a court. Okay. Now, one of the things that is important is looking at how hair grows. And when we think about the hair on our head or on our arms or whatever the case might be, we don't really think about a process. We just think it's always there. And while that is partly true, that's not completely true. There are three distinct phases that our hairs are going to go through. The first one is going to be that of anagen. Anagen is going to be the active hair growth, and on average, this is going to last for about 1,000 days for most individuals. Now, understand that the hairs that we have on our body, regardless of where they're at, are going to be in different parts of this life cycle. So, not all of our hair is in anagen, and then it all goes to catagen, and then it all goes to telogen. That doesn't happen that way, which is good because if it did, then we'd all be bald or close to it, at least at some point in time as we get into that telogen type of phase. But our hairs are growing at different rates, and so this ends up being very useful to us. So anagen, about like I said, the active hair growth about 1,000 days, lots of blood flow to the follicle, so the hair itself is going to grow a lot during this time period. It's going to get to be relatively long during this period of time. And so you can see here that most of our hair at any given point in time is going to be in the anagen hair cycle, or in the anagen uh, phase of hair growth. Then you've got catagen. This is where the blood supply, you'll notice, is decreasing. And so the papilla, remember that papilla is where the blood vessels themselves tie in to the hair follicle and are associated with the hair bulb itself. But that big papilla begins to shrink then, and the hair follicle becomes more club shaped, so it's going to be more rounded in terms of its approximate shape. It's going to last about 10 days, and you'll notice again about 2% of our hair is going to be in that at any given point in time. And then finally that leads us to telogen. Telogen, again, pronunciation is kind of irrelevant now, but telogen is going to be when the follicle ends up being released and the hair can fall out. Okay, and then it's going to have a short period of time where it's going to kind of regenerate itself, and then the process starts all over again. All right. So again, at any point in time, you're going to have hairs that are going to be in each of these different um, phases of hair growth or hair release, I guess, as the case might be as well. Right. So we know that hairs on average for most individuals are going to grow about 1.3 centimeters per month, or about 0.44 millimeters per day. Uh, so again, that's what it ends up rounding out to be in general. All right. So again, um, that is something that can be used by examiners to work backwards to figure out maybe when a person was exposed to a certain chemical, or how when maybe if you have someone that is going in for a drug test and they do a hair sample, when that individual last used a certain type of drug based on the compounds that are present. So again, lots of value in each of these different tests. Again, the way that the hair ends up being removed can help indicate if a crime has occurred. Um, by whether or not a hair has been torn or broken cleanly or cut cleanly, right? If it's been cut with scissors or something like that, versus if it's been ripped out or torn or it's broken or frayed or it indicates some damage has been done to it prior to being removed from the individual. Now, like I said, animal hair and human hair are going to be very different in terms of their appearance. With this, the melanin, like I said, is going to be solid, right? So for animals, their medulla, and I know the melanin's in the cortex, not in the medulla, but in animals, the medulla is going to be continuous. Okay, they have that nice dark line that runs the entire duration of the center portion of their hair. But you'll also notice that any pigmentation is going to be much denser, and it's going to be much more numerous in animals than what it is in humans as well. The medullary index, right, is going to be greater in animals. So if it's zero point 0.5 or greater, if you find a hair where the medullary index, which is the ratio of each of these, the medulla to the diameter of the entire hair, if it's zero point 0.5 or greater, that's going to indicate that it is in fact animal hair and not human. Okay, so again, that zero point 0.5 or greater. And then the cuticle in animals is not going to be flat and narrow. That's what we have, very flattened and very narrow cuticle. In animals, it tends not to follow that same pattern. It tends to be more round and much larger in general. Not always, but oftentimes. Okay. So when we start talking about being able to test hair from a forensic standpoint, again, at the end of the day, the value in it is in what we can show or support with scientifically accepted and vetted processes. All right, so that's what these are going to represent. So one of the easiest things is going to be that of the comparison microscope. And that's just simply being able to take two hairs, put them side by side, look through both eyepieces and see, do those hairs, do they match? Are there pieces? Is the cortex about the same thickness? Is the medulla about the same thickness? Do we see a similar type of medulla pattern from or in both of the hairs that we're comparing. The cuticles, are they the same? Do you see the same type of scale patterns from on both hairs? Right. So that's what comparison microscopy is going to do for us from a forensic standpoint. Again, this isn't going to be able to individualize. So even if you find two hairs that basically look the same, you can't say with 100% certainty they're from the same individual. You can say, hey, the characteristics themselves are going to be very, very similar between the two. And so again, that still has forensic value, just not as much as obviously the individualized evidence that we can get from the DNA stuff. Now, some of the other things that can be done, there are other types of microscopes that allow you to be able to use different light sources and different wavelengths of light, all of which can be very, very useful from a forensic standpoint. Uh, so if you were to have hair that's been treated with a certain type of chemical and you shine a certain wavelength of light on that hair, it may fluoresce or it may give off a different color. And you might have to use different types of filters to be able to see that. But again, forensic scientists are trained to be able to know, hey, this may or may not work, or just to be able to see whether or not they get anything. And with all of these, right, this is non-destructive. Comparison microscopes, non-destructive. Phase contrast, fluorescence microscopy, and even electron microscopy. Excuse me. None of those are destructive. So the hairs themselves can be reused for other tests as well. So that ends up being obviously very, very useful. The neutron activation analysis. what's going to happen is they bombard this a hair sample with um, these neutrons. And as a result, what you get from that is different gamma radiation levels that are going to be released, and from that you can find out whether or not the concentration or what the concentration of certain elements in that sample are. So some of the things that show up in there are going to be antimony, argon, bromine, copper, gold, manganese, silver, sodium, and zinc. All right. And so from those, they can then look at the relative amounts of each of those. Now, this gets to be a lot more specific. Again, so the statistics tell us that the likelihood of two people having the exact same concentration of just nine of those different elements is about one in a million, right? So again, you can start to quantify the likelihood, the probability of seeing certain types of outcomes based on kind of what these chemical tests would be or what that test would be. So again, very useful to give a quantifiable amount. Now, if you are fortunate enough to have the follicular tag or the skin tag, so maybe it's been pulled out. Maybe that skin tag was removed forcibly. Maybe it just came out during antigen somehow. That's not terribly common, but it can happen. Um, then you can have the opportunity to run DNA analysis um, on the cells that are part of that skin tag. And so from that now, you can get a DNA profile. DNA profiles we know are not going to be just that of um, class evidence, but this is going to provide us now individual evidence. All right. So again, only present in hair that was removed during antigen. So once you have that, now you've got individual evidence that's going to be even more specific. All right, so that is where we will end our discussion on hair analysis, again, as the first of our pieces of trace evidence. Next, we will get into fibers and textiles, but we'll save that discussion for next time.